Hello everyone, my name is Ira Chen, a grad student from Virginia Tech, working with Dr. Jing Chen in the Department of Biological Science. Today I'm going to present a poster about my research project on mathematical modeling of mechanosensing and contact-dependent motility coordination in Myxococcus xanthus. Firstly, let's talk about the background of mechanosensing in Myxococcus. For those of you who are not familiar with Myxo, it's a rod-shaped bacteria that reverses its moving direction as it glides on substrate surfaces. This bacteria can sense the mechanical cues in the environment and modulate its reversal frequency accordingly. This is critical for the intercellular coordination and interesting social lifestyle of Myxo. However, the detailed molecular mechanism for the mechanosensing-based motility control in Myxococcus remains unknown. Here we hypothesized that the gliding motility machinery can serve as the mechanosensor and translate mechanical cues into subcellular motility control signal that regulates cell reversals. Here I'm showing you the core model that is a basal model in our study. We have several key components listed by the side of this model scheme. I don't have time to explain all these details, but most importantly, the model integrates the dynamic of the gliding motors with an existing model for the cell polarity control published by Martin Howard and Tai Miao. Then we utilize our model to make two mechanosensing predictions, one about the substrate stiffness, the other on the cell-cell contact stimulus. Based on the core model, we also added motor-phase interactions. This hypothesis is based on the observation of anti-localization of free CD and the motility machinery. Because we don't know what the actual interplay is, we tested all possible interaction between motors and the freeze, and when you compare our model predictions to experiment evidence, you can find out only one mode of phase-motor interplay, which is model 3 here, can generate the observed dependence of cell reversal frequency on substrate stiffness. To stimulate cell-cell contact, we use the transient change of motor engagement rate to represent the effect of cell-cell contact, because the contacting cell essentially provide additional substrate to engage the motors. Therefore, we transiently increase engagement rate for several minutes at different times. You can see that in the first case, the cell reversal is significantly brought forward, and in the second case, there is no significant change in the cell reversal time. This means the first stimulus is in the sensitive period, and the second stimulus is in the refractory period. When simulating with more stimuli over one reversal period, we can achieve the phase shift curve where the refractory period is located right after the cell reversal and the sensitive period right before the cell reversal. Then next step, we plugged our predicted reversal response into a previously published population model to see whether the cell population can form the rippling wave. We find the rippling wave can emerge if cell-cell contact induces sufficient transient increase in motor engagement rate. To sum up, we identified the proper interactions between the gliding motors and the reversal regulators can enable the observed mechanosensing-based motility control in Myxococcus cells and support rippling wave formation in the cell population. Most importantly, the take-home message from our work is that the gliding machinery of Myxococcus could function as the mechanosensor and translate mechanical cues into motility control signals. Thank you very much for your listening.